Okay, good afternoon. Welcome to the second webinar series of the Origin Center of the Netherlands. The Origin Center is a Dutch initiative to bring scientists from different fields together to work on big questions related to origin of life, evolution of life, potential for life, origin of life, evolution of life, and elsewhere. And today's first speaker of this year, of this new seminar series, is Professor Dave Deamer. And we're very happy to have him back to back with Mike Russell, because now we have the opportunity to look at two different origin of life hypotheses. So we start today with Professor Dave Deamer. And he approaches the origin of life question from a biomolecular engineering background. So he really has studied the origin of life from a biomolecular perspective. And today he will tell us from his perspective about his hypothesis of the origin of life. I'm very happy that you're here. Professor Deamer, thank you very much. And with this, I give the floor to you.
Thank you, Inga, for that nice introduction. I want to reiterate that your audience is going to hear two different hypotheses. Mike Russell has been developing the idea that metabolism could originate in the hydrothermal vent environment, and he's extended that along with his colleagues Nick Lane, Bill Martin, and Laurie Barge to the idea that this is a hydrothermal vents are conducive for life to begin in a submarine environment, in a salty ocean. As a biophysicist, I had some concerns about salt water in particular as a place where self-assembly processes can go on. So my research for really my entire career has dealt with self-assembly of molecules like lipids into membranes, like nucleic acids into their double helical structure and hairpins, and RNA as a catalyst, so-called ribozymes. So I've been going on a different tack than the hydrothermal vent hypothesis. And I think this is a healthy way for science to advance, to have alternative hypotheses which are then open to experimental testing. So for my talk, I'm going to be telling you about experimental tests that we have done with a hydrothermal field idea that a freshwater hydrothermal field is at least equally plausible as a site for life to begin and does have some advantages. So I'm going to emphasize some of the advantages in my talk. Mike is going to emphasize the advantages of hydrothermal vents in his talk. And then you are kind of like a jury, and you're going to decide on which hypothesis has more explanatory power. Right now, we don't know what the future holds, but right now you should be able to have your own opinion on explanatory power. So I'm going to begin with the history of my involvement with this. Here you see three examples of hydrothermal conditions. The two on the left are the black smokers discovered by Jack Corliss in 1977 while he was diving near the Galapagos in the submersible called the Alvin. This was quite a surprise to find these extraordinary smoking vents, many thousand meters deep just off the Galapagos Islands. Just a couple of years later then, Jack and his colleagues, John Burroughs and Sarah Hoffman at the University of Washington, published the first suggestion that life might have begun in hydrothermal vents, particularly like these black smokers, because the fact is these are loaded with living organisms, all the way from microorganisms, bacteria, and so forth, taking advantage of the energy available at these black smokers as well as larger organisms like the tube worms and certain crustacea that have found a way to make a living down there. In the middle are alkaline vents. Now, these were discovered by Kelly et al. Off the coast of North America, and these are not fed by the heat of magma like the black smokers are, but in fact by serpentinization, which is a chemical reaction between seawater and olivine. The result is a very alkaline fluid high in hydrogen, dissolved hydrogen gas. And this is what the hydrothermal vent people are now talking about as a conducive site. These are lower temperature. These are down in the ordinary sort of temperature range, 70, 80 degrees C. And they also are inhabited by microorganisms. So this is what Mike is going to be telling you about primarily, the alkaline vents and the kinds of energy that are available there to give rise to a primitive version of metabolism. Clear to the right is one of my favorite places. This is Bumpus Hell on Mount Lassen in Northern California. And we've been there half a dozen times, both exploring the area as well as doing field work. We're trying to do some experiments there. And toward the end of my talk, I'm going to be telling you about some experiments we've been doing in New Zealand in a hot spring area, similar to Bumpus Hell here. This is fresh water. These are millimolar concentration of ions. They are fed by precipitation, rain and snow that keep the water there. And these are at near boiling temperatures, around 90 degrees C. They're also somewhat acidic, about pH 3, which is the acidity of lemon juice, for example. So I think all of those will come into play as I get a little further into my talk. Now I want to show you one last thing here, and that is keep in mind that the submarine hydrothermal vents are in salty seawater. This is half molar sodium chloride, about 50 millimolar magnesium, a divalent cation, and 10 millimolar calcium ions in solution of seawater. So that's what is allowing these structures to form, because the highly alkaline material reacts with the calcium in the seawater and forms calcium carbonate minerals, and that produces these towering mineral structures you see. Alternatively, the hydrothermal fields are low pH. They are very low concentrations of cations. Sodium chloride, for instance, might be about 10 millimolar. Calcium is down below millimolar, and as is magnesium. There's almost no magnesium in a typical volcanic hot spring like this. So that's important to this story that I'm going to be telling you. Freshwater versus salty seawater. So this story begins with 2 trips to Kamchatka that I made in the maybe 15 years ago now. So I want to show you this trip, just a few slides, because this is the basis of the ideas that I'll be presenting today. Now, to get to Kamchatka from San Francisco, we hop on a plane up to Anchorage. We stay overnight there, then hop on a Russian plane, Magadan or Airlines, which takes us over to Kamchatka Peninsula. And just below Kamchatka, by the way, that's where Japan starts. So Kamchatka and Japan are very volcanic, as you know from just watching Mount Fuji erupting in Japan. Kamchatka is amazingly volcanic, a whole string of volcanoes starting down in the south part near Petropavlovsk and going all the way up the spine. And you can see these volcanoes in this artist's view. This is taken from a satellite image, and all of those white mountaintops are in fact volcanoes, a string of volcanoes. Now, if you look off to the right of that slide, you'll see Petropavlovsk, and that is where we land after we get done with the trip from Anchorage, Alaska. And then we have an overland trip down to Mount Metnosky. And Mount Metnosky is an active volcano. Oh, by the way, this is what Petropavlovsk looks like. Petropavlovsk sounds very Russian, but it's just the words St. Peter and St. Paul. And they were named by the early explorer who discovered the bay of where Petropavlovsk is now as a city. It's a beautiful little city. Maybe 100,000 people live there. This looming Koryatsky volcano is about 50 miles away, and that just is the nearest volcano to Petropavlovsk. So we hop on a Russian troop carrier, which is now turned into a tourist guiding vehicle, and they took along two of our little groups in a couple of years apart. And we then travel this dirt road south from Petropavlovsk, and you can see that white line. Then Mount Metnovsky is right there. That is this active volcano. It erupts every few years. It's really quite a place to visit, as you'll see. We then travel overland from that road to near the base of the volcano. And there's a little wooden shed there that we can stay in for about a week's time while we do our work. Here's one of our trips. This is my last trip there. This is a group of scientists. The two women are grad students at Stanford University. They've both gone on to later careers. The guys in the red and the black jacket are Jan and Jake. They were postdocs at Carnegie Institution. The person in the white jacket is our local organizer. He's a volcanologist, Vladimir Kompenichenko. And if you ever want to visit the volcanoes, Vladimir is the guy to see. He's a scientist, and he just loves to help people discover these remarkable landscape of Kamchatka. Clear to the right is Igor. He is our guide for Mount Metnovsky. And clear to the left is my neighbor, Tony Hoffman, who is the unofficial photographer on the trip. He just lives down the street from where I'm here in California. So here we are next day, traveling up a ice field to get to the crater of Mount Metnovsky. Here is Vladimir looking down into the crater. And this is what I imagine when I think what the early Earth was like. We call this a prebiotic analog site because it's a place where we can get pretty close as can be, to what the Earth was like 4 billion years ago. So that's why we like to go to these volcanic sites, because we can do experiments there. And it's kind of like visiting a prebiotic Earth and seeing what happens as organic molecules react with the conditions that would have been abundant on the early Earth. So here I am now down in the crater. This area has now been buried by a recent eruption, but I was taking a sample out of this fumarole, and I took the sample back to my lab in Santa Cruz and discovered that almost no organic compounds were present in this hot, boiling mud of this fumarole. By the way, you see that yellow stuff around the fumarole, that's elemental sulfur that just comes out of the gas phase emitted by the volcanoes. That smoke that you see is mostly water vapor, but it also has hydrochloric acid in it, hydrofluoric acid, believe it or not, SO2, which is sulfur dioxide, and that's a pretty toxic mix. So once in a while, we had to quickly put on gas masks to protect ourselves when the wind blew this toxic gas in our direction. But I knew that we probably would not find any organic compounds in this. So I brought along some organic compounds and looked around for a small pool to do an experiment. So this is a boiling hot spring. It's enclosed, it's just a little puddle. And the murkiness is due to clay, which is suspended by the boiling action. And the bottom of the pool, in fact, is sealed by the clay that is being produced from the basaltic minerals that surround this particular pool. So the idea was, what would happen if I put in a mixture of molecules, organic solutes, that would be like what we think could have been available on the early Earth? So that white material I'm pouring in is a mixture of amino acids, glycine, alanine, valine, and aspartic acid. All of these we're pretty confident were on the early Earth, because even today, these are being delivered by meteorites, such as the Murchison meteorite that I'm going to tell you about a little later in my talk. 4 nucleobases. These are the nucleobases of ribonucleic acid: adenine, uracil, guanine, and cytosine. And I wanted to know what would happen to actual nucleobases, the main molecules of nucleic acids. Then there's an amphiphilic molecule present, a 14-carbon fatty acid, and that's very important to the story that I'll be telling you. That made it a white, milky suspension, because fatty acids can form microscopic membranes when you are at the right pH. So this little beaker then has water from that pool, because these were all dry to begin with. I picked up the water from the pool, just stirred it around, and poured it back in. 2 other things that were present are glycerol and phosphate, in case I could see any synthesis of a phosphatidic acid. This is one of the lipids that can form membranes. And then again, it's a low pH. So what I saw immediately within minutes is a white froth appearing around the edge. And that white froth, of course, is this soap-like molecule, that fatty acid that I added. And it boiled to the surface of the pool and then was pushed to the edge. And that is where this idea I'm going to talk about came from. This pool isn't eternal. It goes through cycles of hydration and dehydration. It evaporates around the edges. Sometimes it completely evaporates. It gets refilled by rain and snow melt. And these cycles then, I think, are very important to how life can begin. Because during the dry phase, chemical energy is made available to drive polymerization. So the idea that I'm going to tell you about basically started here in 2004 with the understanding that this kind of a pool could have driven some prebiotic chemistry had not been previously explored. Everything else that I added, by the way, was adsorbed to the clay. It disappeared from the solution and no longer would have been available to react to produce polymers, for instance. And this is something unexpected. The cytosine I added, by the way, was not stable. It was deaminated and turned into uracil. So when we analyzed this water back in the lab, instead of 4 nucleobases, there were only 3. This was published, if you're interested, a couple years later. And that's, again, my little pool in Bumpus Hill, the cover of this philosophical transactions. And all of the detailed chemistry that we discovered is in the article that we have here. So let's go on now. What are the favorable properties of volcanic hydrothermal fields? And these are in contrast to the properties of hydrothermal vents. Organic compounds would accumulate in hydrothermal ponds, and that's very important. If they are produced, they will tend to get flushed into those ponds and thereby undergo concentration by wet-dry cycles. The ionic concentrations, in contrast to seawater, are really low, down in the millimolar range. So there's almost no calcium, magnesium, a little bit of sodium chloride, and that's about it. And we've looked at pools all over the world now, and that's generally true of volcanic hot springs. Wet-dry cycles during the dry phase concentrate potential reactants. That's very important. Polymerization, we now know, is promoted in the dry phase of a cycle. And you're going to hear a lot about that because we've tested this idea experimentally. These places are hot, 80 to 90 degree water. 89 degrees Celsius, of course, near boiling, and that provides activation energy for the chemistry. Membranous compartments. If lipids are there, they will assemble into membranous compartments and capture polymers to form protocells. And those 6 points are, in fact, the fundamental basis of the ideas that I'm presenting to you. That goes from beginning to end, starting with simple chemicals and ending up with protocells containing polymers that are synthesized during the wet-dry cycles. So where do the organic compounds come from that I need for this to work? Well, from meteoritic infall. That's one primary source of organic chemicals on the early Earth. We know that because they are still being delivered today. What I want to show you is a couple of slides from other laboratories that have worked on this. This shows the cycle all the way from the dust in the solar nebula around the early sun. And that dust is in fact minerals, microscopic minerals of olivine and pyroxene with an ice layer on the surface. That's really where a lot of Earth's water comes from, because this stuff accumulates in planetesimals and comets and then is delivered to the early Earth during accretion. So that ice layer, we know, when exposed to ultraviolet light, undergoes photochemical reactions to produce amino acids, purines, hydrocarbons, and amphiphilic compounds, such as that fatty acid that we put into the pool. So that is a source coming in on what we call now interplanetary dust particles. But back then, those are dust particles with these organic compounds and water surrounding the early sun as planet formation goes on. Those dust particles accrete into comets, planetesimals, bringing the organic compounds along, and those are delivered to the early Earth. Now, here's a paper that I really was impressed with when I first read it. This is by Chris Chiba and Carl Sagan, published in 1992, and I recommend this reading if you're not familiar with it. Chris was a very well-versed in computational approaches to delivery of organic compounds, and he looked at interplanetary dust particles, airbursts, which is what happens when a big impactor hits our atmosphere, cometary impacts, and meteorites. And these are, look off to the left, delivery of infall exogenous organic compounds. So this is the stuff that is being delivered. And if you look at the time scale down below, you can see 4.0 billion years ago. Bring your eye up to IDPs, and you see that it's something like 10 to the 8th kilograms of organic carbon per year. And the IDPs are the primary source of organics being delivered to the early Earth 4 billion years ago, right at the time that life was starting up on the Earth. If you divide that number of 10 to the 8th kilograms per carbon per year by the number of square kilometers on the early Earth and the Earth today, that's about 10 to the 8th square kilometers. So we are finding then, or estimating in this estimate that Chris and Carl came up with, that about a kilogram of organic material was being delivered per square kilometer per year. So when you add up millions of years that this stuff is raining down, you can see that it becomes pretty significant source of organic material. So that's what I hang on to. I think this is reasonably certain that organic material was being delivered because we still see it being delivered today. So here's an illustration of what I just told you. We have the interplanetary dust particles on the in the solar nebula surrounding the sun, 4 billion years ago. It's being delivered to the Earth, accumulating on volcanic land masses. It gets fleshed into hydrothermal fields, basically the sort of hot springs that I showed you today in Kamchatka. And those hot springs evaporate and refill, cycling this stuff through indefinite numbers of wet-dry cycles. Here's a little movie that we made, done by a very talented Australian videographer. This is what we think the Earth might have looked like 4 billion years ago. Nothing alive, but we do have geyser activity. We have hot springs on volcanic islands coming up out of the global ocean. That brown stuff you see on the surface is organic material that is accumulated, is being flushed down into all of these pools like Kamchatka and like Bumpus Hill that we've seen. And this water level goes up and down, both by evaporation and rehydration, but also just by like a bathtub ring forming as this water goes up and down against surrounding rocks. So let's take a look now at one example of how organic compounds can be delivered. This shows an agricultural field near Murchison, Australia. And this would have been a summer day, late summer, September of 1969. And the people of Murchison looked up in the sky at a fireball coming in, which exploded, and pieces of the meteorite fell all over the fields surrounding Murchison. Each of those red dots is a chunk of the Murchison meteorite. Something like 100 kilograms were quickly picked up, both by townspeople and by scientists who rushed to the scene to capture this fresh infall. These are available for distribution to scientists around the world. I've had 3 chunks of Murchison come through my lab. This is one of them. I think this is the first one. It's about a golf ball sized object. You can see the fusion crust on the surface just because of the ablation that occurs as it moves through the atmosphere. It gets very hot, but the interior stays pretty much at the interior temperature of outer space. Carbonaceous meteorites contain organic compounds. Look at that. About over 1% is an insoluble polymer called kerogen. About 0.1% of the mass of the Murchison meteorite is soluble organics, and those are very interesting. Half of those are carboxylic acids, including soap-like molecules up to about 13 carbons in length. If you look at the other technical names there, we have dicarboxylic acids, about 50 parts per million of amino acids. There's amides there. There are oily hydrocarbons, aromatic hydrocarbons, aldehydes, alcohols, amines, and even purines. We now know that purines like adenine and guanine are among the compounds present in the organic mixture of the Murchison meteorite and in other carbonaceous meteorites, but the Murchison is the best study. So this is what I hang on to, as that this is a source of the kinds of molecules required for life to begin. They're being delivered still today, and 4 billion years ago, they were being delivered thousands of times faster. So life began by assembly of certain kinds of organic molecules. Nucleotides are monomers of nucleic acids. Amino acids are monomers of proteins. And amphiphilic compounds, lipids, those fatty acids I talked about, can assemble into membrane compartments. Life is cellular today. It probably began as primitive cellular structures. We call these protocells. So membranes are composed of these amphiphilic molecules. 2 of them are shown here. The first one is a fatty acid. That's got about 16 carbons in it. That's a fatty acid called palmitic acid. And this is ordinary soap molecules. And yet we can find things like this in the Murchison meteorite and other meteorites. soap molecules have this wonderful ability to self-assemble into membranes. And probably everybody listening to this talk at one time in your early life blew soap bubbles. You were making membranes self-assembled from fatty acids such as we see in meteorites. Membranes in cells today are bilayers, lipid bilayers. They have a hydrocarbon interior and hydrophilic groups on the outer surfaces of the membranes. And you can see water molecules, the H2O, in this little illustration of a lipid bilayer. So when I talk about a membrane, keep that in mind as the essential structure of all life on Earth today, and presumably at the beginning of life. Membranes would have encapsulated polymers to make the initial step towards cellular life. Now, this is interesting. Back in the 1980s, I extracted material from the Murchison meteorite, put it under the microscope, and some of the compounds assembled into the membranes you see here. So these things are over 5 billion years old because they were present on the dust particles of the clouds that around the early sun of the solar system. These were delivered to the Earth in 1969, and here they are still able to assemble into membranous structures. We know that they are compartments because we can encapsulate a fluorescent dye inside. And over to the right, you can see an encapsulated dye fluorescing as it's encapsulated in those vesicles. So that was very, very encouraging that in fact membranes were very likely abundant on the early Earth. They self-assemble spontaneously and they would be available for the first forms of cellular life. So something important that you need to know, because this is part of what the story I'm telling is that membranes can encapsulate small and large molecules related to life. When dehydration of lipid vesicles occurs, those membranes that you see there undergo a fusion in the dry phase, and they form what we call a multilamellar matrix, encapsulating those little round things, which could be anything you want, like that fluorescent dye, or these could be the monomers of nucleic acids. When you add water back, you discover that in fact you have encapsulated up to about half of the molecules there. Here is a fatty acid. This is decanoic acid. And any of you could do this in the lab. If you have some DNA and some soap molecules, just dry them down together, rehydrate, and look at them under the microscope. You will see that you have made what we call protocells, a membrane encapsulated DNA. The DNA has been labeled with the fluorescent dye so that you can see it inside these fatty acid membranes. And that's all that's there, decanoic acid, simple fatty acid. So now we're getting to the last part of my talk. How did nucleic acids first assemble on the early Earth? First, we need what is a source of nucleotides. Well, I've got to say right off, we don't know for sure. It's not like nucleotides are available in the same sense that amino acids are available. Powner and Sutherland, however, have published some nice organic chemistry showing that at least it's conceivable that nucleotides would be available. So we just take it as a given. If you're going to make nucleic acids, you've got to have nucleotides available. Then you need to know what free energy drives condensation reactions. How can the entropy barrier be overcome? It's a big mix of stuff. It's all disorganized. How can you make it more organized so that polymerization can happen? How can nucleic acid resist hydrolysis? Because if you make something, it goes uphill by removing water. If you add water back, it goes back downhill to nucleotides. So that's an important aspect that we have to understand. Finally, how can nucleic acids evolve functions such as catalyze growth and replication? This is the last step toward the origin of life. I can answer those first 4 questions based on laboratory experiments that I'm going to tell you about. We don't know yet that answer that question number 5, how evolution can begin. So here's the deal. We're going to try to simulate hot springs in the laboratory. We're going to set up a bunch of conditions, a carbon dioxide atmosphere to make it anaerobic, elevated temperature, acidic pH range, and hydration, dehydration cycles. So what happens when nucleotides are exposed to those conditions? Here is a little apparatus we built. It's a simulation chamber. We can fill it with carbon dioxide from that big tank off to the left. We have water coming in, cycling in, and little vials are rotated under gas, CO2, over a heat source with periodic hydration every half hour, for example. So go through wet-dry cycles, then we can see what happens. Now, here's something you've got to know. It's simpler than you think to make nucleic acids. For instance, if you mix ethanol and acetic acid in the laboratory, they spontaneously form a certain amount of ethyl acetate by making an ester bond. That's called a condensation reaction. Water is a byproduct of this. Now then, here's the trick. The same thing can happen with nucleotides. Here's adenosine monophosphate. It's one of the 4 nucleotides in DNA and RNA. An ester bond, by the laws of chemistry and physics, can form between the phosphate group and a hydroxyl group on the ribose, on the next neighboring molecule. And you can see the ester bond. DNA and RNA are polyesters. I'll bet you never thought of that when you buy a polyester shirt. DNA and RNA are also polyesters. Water comes off as a byproduct of this. If we remove the water, somehow you can pull the reaction to the right so that you make more and more of the ester bonds. So that is what we do when we dry out a solution of mononucleotides under these simulated conditions. We expect to have ester bonds synthesized, and as you'll see, we actually see that happen. So here's 6 tests I'm going to run. These are in the Kuhnian tradition and the idea of falsifying your hypothesis. These tests are an attempt to falsify this idea that I'm passing on to you. So here's test number 1. We should be able to have mononucleotides. We can have lipids there if we want, multiple wet-dry cycles. We should be able to precipitate the products by ordinary precipitation methods used in molecular biology. And if we have made polymers, they should be able to be labeled by radioactive phosphate. We can then do gel electrophoresis. And here's a paper that we published now, 2008, with Sudha Rajamani as first author. You can do this with any RNA you want. You have 2 enzymes called alkaline phosphatase and T4 kinase, and they can add a radioactive phosphate to the end of RNA molecules. So if we have made RNA by those conditions, we should be able to see it in gels. And there they are. These are all different experiments. Each of those lanes is a different experiment, and you can see that this is a very robust reaction. We get polymers able to be labeled with a radioactive phosphate ranging from about 10 mers all the way up past 100 mers. And those are all different conditions, different nucleotides, different lipids, and we are able to see these labeled polymers. So the first test succeeded. Next test, can they be seen by fluorescent labels? The answer is yes. If you look on that left-hand gel, you'll see nothing happening with poly U or poly A. Those are synthetic RNAs that should be able to form a double helix if you combine them. So look They're poly A plus poly U. You can see that now they're labeled with ethidium, an intercalating dye that can get into double helical structures. And there's our product, clear to the right. That is that same product we saw with radioactive labeling, and it's labeled by these dyes. Then off to the clear to the right is another dye, another kind of a gel, and you can see the product again. So we're quite certain that we are making something resembling RNA. Well, what about this? Here's test number 3. We should be able to sequence those polymers using nanopore sequencing. Nanopore sequencing is something that was developed here at UC Santa Cruz. You can now buy nanopore sequencing devices from Oxford Nanopore Technology. It works by having a helicase unwinding a double strand that gets pulled through a pore, what we call a nanopore, embedded in a membrane. And as each group of bases gets to the portion of that pore where there's a constriction, it produces a change in the ionic current. And those are base-specific changes. And you can see those modulations in that little square on the right. So to test this out for the first time, this is unpublished, so you're hearing this unpublished results. What we did was take thymidine monophosphate, which is a monomer of nucleic acid of DNA, not RNA. We went and put through multiple wet dry cycles. We expected to get oligothymidyllic acid, just the polymer of the TMP. We then ligate to a known DNA because the oligos are too short for the nanopore sequencing. We have to have something around 500 nucleotides long. So we just ligate this to a known DNA. Then we do nanopore sequencing. To do nanopore sequencing, you just inject your solution into that little device about the size of a cell phone. It feeds into your laptop, and you can actually watch as single molecules go through something like 500 nanopores in this little instrument, and you can watch the sequences turning up on your computer screen. If you look at a single strand going through the pore, this is what you see. It goes into the left strand entering the pore. All of those ups and downs are the base modulated ionic currents in the picoamp range. Suddenly, it drops down to a long strand where the oligo DT gets into the pore, and then it comes out clear to the right-hand side. So if we do the base sequencing of these sorts of signals, we see this. First, there's all of our oligomers. You see them radioactively labeled. There's the gel clear off to the left. And then in that white on black, you see a scan of the gel with each of those separated by the electrophoresis. Finally, we see the base sequence. And there, time and again, we see the oligo DT attached to the rest of the DNA. This is really convincing if you believe nanopore sequencing, because these can be correlated with shorter strands like 30 mers, with longer strands down in the 40 mer range, and with really long strands up in the 80 mer range. So that's the kind of evidence that we have. Test 4, we should see these encapsulated by a wet dry cycle. This is just a simple solution of decanoic acid with decanoyl monoglyceride. We put it through 4 cycles and looked for DNA by a DAPI stain, that is fluorescent stain. And we can see the DNA accumulating in the cell, in these protocells. After 4 cycles, you see nice little gatherings, aggregates of the DNA that's been synthesized in the cell. This is a surprise. That's where we visited last year. It's a hot spring area in New Zealand near a place called Rotorua, little village area in the North Island. This is called Hell's Gate, and it's a very active boiling hot spring area that is something of a tourist attraction. We were given permission to do experiments there by putting mixtures of AMP and UMP into those hot springs with hot spring water to see whether these laboratory experiments would actually work in a hot spring area. We did this twice now, in 2018 and in 2020, just before the pandemic hit. We got back in February of 2020, and we were able to analyze this in the laboratory. And you can see these are micrograms of product precipitated out of those waters after 4 cycles, 4 wet-dry cycles. And we got a similar result in 2 different attempts. If you look down at the bottom, you can see that the uracil containing mononucleotides dominate the micrograms in both cases. The adenine, a little bit less yield, but they're up near the top of those things. We finally did gels. I'm getting right toward the last few slides of my talk. We can see the product in the laboratory. We see the same product off to the right in the hot spring conditions. Finally, can we visualize the polymers? This was being done in Copenhagen, Denmark, with my colleague Tue Hassenkam. And this published last year. We can see little ring-like structures being produced as we use atomic force microscopy to look at the polymers we're making. These are several hundred nucleotides in length, and they're very common. You can see them. We just point at the larger ones. So we have a lot of evidence supporting the idea that we can really get polymers made. There's a little collection of the rings that we're seeing, both with GC and with AU mixtures. Single nucleotides do not form rings. We think that's a clue to how this polymerization can work. And I'll be happy to talk about that. So to summarize, here's an alternative scenario, freshwater origin. And you know all about this now that you've heard my talk. Life began in freshwater hydrothermal fields. Wet-dry cycling is necessary. Life did not invent nucleic acids. Instead, the chemical energy made available by wet-dry cycling generated random RNA-like polymers. If amphiphiles are also present, they can form membranes that encapsulate the polymers in vast numbers of membranous compartments we call protocells. Each of these is a microscopic experiment because they are all different. We make trillions of these every time we do an experiment. They're all different in size and the composition of the polymers. So we think that a few of them can begin to undergo selection and evolution, because even though most of these protocells are inert, they'll be discarded and recycled, a few of them may be more stable. They might have properties that will allow them to survive further selection. So life began when selected systems of encapsulated polymers emerged that were capable of catalyzed metabolism, growth, and replication. And that can occur in a place like this. We have a synthesis of organic polymers on dust in the dust particles of the protocellular nebula. It's delivered by infall. It accumulates on volcanic land masses. It gets cycled in hydrothermal pools, and the result are these protocells that I told you about. Okay, that's as much as I have to say. I'll now get out of slide sharing and take any questions you may have.
Thank you very much for this very inspiring presentation. So I would like to invite the audience. Are there any questions? Yeah, thank you for stopping sharing. Are there any questions from the audience? Yes, Eloy, go ahead, please.
Hi. Well, I just wrote these things because I presume the talk would go on until 4 or I guess 5 p.m. here where you are. I'm in the Canary Islands next to an ongoing volcanic eruption. So maybe David would like to come here. There's not a lot of hydrothermalism here. It's just lava pouring out.
Well, I was just going to say, if you give it a few more years and some rainfall, you'll get these hydrothermal pools.
Oh yeah, for sure.
Just wait for it to cool down.
Yeah, no, I just wanted to point out because at some point, I mean, I guess everyone, as we say in Spain, everyone pushes things towards their own yard. So of course, David does that, I do that, and everyone in the field or any other field does that. But I would like to point out that some of the features that David and many others find interesting about superficial submarine systems, at least some of them are also present in submarine and other types of systems that are neither of those, such as heat is very prevalent in the early Earth. And also recently we've known that high salinity is actually not bad for assembly of some type of membranes, depending on the fatty acid or fatty alcohol composition of them. And that also organics can accumulate in the sea, believe it or not, as long as you are in a porous system where a physical phenomenon known as thermophoresis can take place. So, you know, we all look at these properties, but they are, in most cases, not exclusive to one system or another.
Yeah, that's a good point. I know about the accumulation of nucleotides. Were you an author on that paper showing accumulation in porous minerals?
I am authoring one of them, but yeah, there's many, so I don't know. I know that Dieter Braun in Munich has done quite a lot of work on that.
That's right. That's right. So what I would like to see is, you know, these are simulations you're talking about, and it's not actual vent minerals. I think it'd be really neat to try to get somebody to gather up some of the vent minerals, either from the black smokers or one from the alkaline vents, and see if this accumulation works in a true porous vent mineral. Yeah, I think that's essential to try, because the claim and the demonstration that you have this really remarkable I guess you call it thermophoresis, accumulation in porous minerals. I think that's a very valid observation. But let's try and get it to see whether it actually works in a known vent mineral. That really put the nail.
We did it indeed in 2014 in alumina pores that we created. What we found out though is that it was quite tricky to study, because every time you made these structures, the pores and tiny channels were very randomly distributed. So every time you would find like crazy accumulations, like 3,000-fold and above. But they were always in different places and in different amounts. So it was very hard to like standardize. So I think that's why Dieter then went on into all of the simulations just to see what would happen in one perfect pore. But of course, these are all simplifications.
Yeah, I think another possible mineral that is easy to get lots of is pumice.
Oh yeah, indeed.
So this would be one that you could try as an actual volcanic mineral, porous mineral.
Good idea. Thanks. That's it.
Yeah.
Are there any other questions?
I would like to suggest a question that I won't be able to answer. We know for a fact that chirality is a very important aspect of life today, homochirality. And years ago, Jerry Joyce showed that monomeric nucleotides that are the wrong chirality, having L sugars, L ribose, instead of D ribose, you know, they work very well. They don't polymerize very well on a template that they were working with back then. So a hard question for me would be, how do I know that we can get past this problem that the first nucleotides would almost certainly be raceme? It would be a mixture of D and L ribose, for example, in the nucleotide. And we would not expect those to be able to, to be able to polymerize very well. So that's a problem. Of course, everybody has that problem. We just tend to work with what works. And what works is all D ribose in these nucleotides. Another hard question is, how do I get past what is called the 2,5 and 3,5 ester linkages? In RNA, you can have two different linkages between the phosphate and the ribose. It can either be to the number 2 hydroxyl or the number 3 hydroxyl. And whenever you make something without using enzymes, you tend to get a mixture. And once again, these things do not fold up very well into active configuration, such as a ribozyme would need. That has to be really all 3,5 mix of the bonding. So that's another hard one for me to deal with. We're just not there yet. So there's some really good questions. I can also suggest some questions you might ask next at the next talk if somebody wants me to come up with some hard questions for Mike to try to answer.
I see Aloy has another question for you.
Okay. I promise it's the last one, but it's related to this very interesting point about chirality. And we've recently been working in Inge Luce's group. When I was still there, I just left very recently. And we have a way of polymerizing some ATP, I believe, but I think it also has worked under certain scenarios with AMP. And we've been using MALDI-TOF, David, to confirm that we have the masses, and we know they are not adducts, they're actually dehydration reactions. But we don't know exactly, as you say, which OH group is the phosphate group being attached to. How would you find out, which technique would you use to differentiate between 3' OH versus 4 or 2' OH?
Yeah, the easiest is that some enzymes will attack just 3,5 or just 2,5 or both. So that's what Jim Ferris did. And he could sort out not the specific site of a 2,5 or a 3,5 bond, but he could say that the polymers he was getting was like 40% 2,5 and 60% 3,5. Mm-hmm. So that would be one way to get at that question.
I see.
But when I've tried MALDI and electrospray mass spectrometry, these mixtures that we make are so complex. They're all different lengths. They're all different sort of linkages that we just kind of get a big blob of stuff. We know it's a polymer, but we don't get separation into specific classes. Of course, at the shorter oligomers, di 2, 3, 4, 5, 6, we can see those. And Sudarajamani, in fact, has specialized in using the mass spec and HPLC combined to look at those.
I'll check that out.
Yeah. But what has convinced me, or what I thought was very convincing, that when we use TMP, we can only make 3,5 linkages. And you saw that gel. We see a big ladder all the way up to 90 mers of labeled, separated by single nucleotide resolution in this sequencing gel. So that really is suggestive that we are making actual polymers that have 3,5 linkages, because that's the only ones that can be made. So there I would be able to use, GC mass spec, HPLC mass spec to separate those. Mm-hmm. Those are expensive instruments, and several $100,000. And I don't have access to one right now.
Cool. Thanks for the suggestion. We might need to add you in the author list, but we can get in touch in the future. Thanks.
So I have one more question here from Dario Campisi. He's a computational chemist and a computational astrochemist. And he asks, he can't ask live because he's actually listening in a bit a noisy location, but he wonders if self-assembly structures might be driven by molecules that provide RNA structures, such as polycyclic aromatic hydrocarbons. Could those serve as kind of a template? Or
yeah, it's a very interesting point. The polycyclic aromatic hydrocarbons are generally kind of a flat molecule and resemble cholesterol in an odd way. Cholesterol has 4 rings in the structure. Pyrene has 4 rings in the structure. Fluoranthene has 4 rings in the structure. And those are very common, PAH in the Murchison meteorite. And so PAH are going to be a fairly common component and might be able to intercalate into the double strand. And Nick Hudd has found some really interesting chemistry that can go on if you have an intercalating dye, such as ethidium, that can fit between the bases in a double helix, for instance, those are in fact a self-assembled structure, the base structure. You've got these stacked bases. So that does promote stability and it promotes synthesis because it prevents the little nascent structure from cyclizing instead of just turning right around and turning into a cycle, which inhibits further polymerization. It allows the polymers to grow very long. So I think PAH is something that deserves a lot more study as a component, both in terms of the biophysics of what is going on and the biochemistry and chemistry of what can go on in polymerization. I don't know if that got at the question.
Well, he answers, thank you. This is really interesting. So I think you answered his question. So thank you. Okay. I think we have time for one final question. And I see a question on YouTube popping up. There the question is, would your experiments and your theory mean that protocells are being created all the time on Earth? If you look at these pools in Kamchatka, for example, or do we really need prebiotic Earth conditions?
Okay, so if you're questioning the idea that we might see protocells in today's Earth, the problem that is faced by protocells today is that there are ferocious, highly evolved microorganisms that would eat them up. Yeah. Charles Darwin mentioned this in a letter to his friend Joseph Hooke back in 1871. And his intuition is that life could not begin today because we are infested with the microorganisms that are just going to eat anything that comes along that can provide nutrients. Another problem today is the oxidizing atmosphere. Molecular oxygen is a voracious oxidizing agent and really contributes to human aging. We are all the time fighting off the toxic effects of oxygen that we use as an energy source. But boy, some of the byproducts are toxic and certainly part of the aging process of all life. So going back to the early Earth, on the other hand, there's no oxygen. So reduced compounds would be lasting much longer. They'd be sort of protected from the oxidation damage. There's nothing there to eat them. So the worst thing that can happen is they just fall apart again and their parts get recycled. My last point is that what allows them to accumulate is what we call a kinetic trap. The polymers become more and more resistant to hydrolysis, the greater the secondary and tertiary structure that they fall into. They start to hide the hydrolytic sites from water. And double-stranded DNA, for instance, is much more resistant to hydrolysis than single-stranded. Same for RNA. So this is something to keep in mind, that these kinetic traps are very much part and very much essential for the accumulation of polymers uphill away, toward lower entropy states than the downhill reaction of hydrolysis.
Okay, well, thank you very much. This was a very clear answer. And with this, we will end today's live stream. So thank you very much again for being our speaker, our first speaker of this series as well. And I hope to see you all, the participants, again on the 9th of November, when we will have the next speaker, Mike Russell, who will talk about the origin of life at the seafloor. So thank you very much. And we will close the live stream now.